Hi, my name is Adi Kapoor, and welcome to this presentation on McArdle disease. So, what is McArdle disease? Well, McArdle disease, aka glycogen storage disease 5, or GSD5 for short, is a series of mutations on the PYGM gene, and the specific region it's most commonly found on is the R50X region. And this gene is located on chromosome 11. The PYGM gene codes for an enzyme called glycogen phosphorylase. This protein is responsible for breaking down glycogen into glucose in your muscles during exercise. Mutated or ineffective copies of glycogen phosphorylase can result in a variety of symptoms. Some of these include impaired energy production during physical activity, exercise intolerance, and muscle pain. And then just a bit of light history on McArdle. The name comes from Dr. Brian McArdle, who first described the disease in 1951. And then worldwide, 1 out of every 100,000 individuals are affected by McArdle. So what are the signs and symptoms of McArdle disease? Symptoms present in early childhood or adolescence, but typically diagnosis of these symptoms is delayed until adulthood, where they reach their severity. And then these symptoms, being exercise intolerance, muscle pain, cramps, etc., occur after physical activity, meaning these are exclusively physical symptoms, and there's no direct cognitive effect. And then severe cases of McArdle has symptoms such as rhabdomyolysis, which is a condition that has muscle fiber breaking down quickly and leaking into your blood and releasing harmful substances. Another example of a severe symptom in McArdle is kidney damage, and a third one is myoglobinuria, and then what that is leakage of muscle proteins into urine, often turning it dark. So let's talk about the pathophysiology of patients with McArdle. So what pathophysiology is, it's how a disease changes the normal way one's body functions. And then in the case of McArdle, the first would be an overall lack of readily available glucose in skeletal muscles, which leads to the previously stated symptoms. Another way one's pathophysiology is affected is by experiencing the second wind phenomenon. And this is a interesting case where people report an improvement of symptoms after post-exercise resting. What this can be boiled down to is your body is utilizing other sources of glucose and fatty acid when your glycogen phosphorylase is unable to break down glycogen. And then a third way is reduced OGLC NAC elation, which is a post-translational modification where a single N-acetylglucosamine is attached to a serine or a threonine. And just to explain those terms, a post-translational modification is a change made to a protein after they've been built, which affects their function. And then OGLC NAC elation is a small sugar that's added to a protein to aid its function. And then lastly, serine or threonine are amino acids, which are the building blocks of protein, where chemical tags can be added. And essentially what this reduction results in is impaired enzyme activities. Sort of to re-emphasize it, lower rates of OGLC NAC elation reduce enzyme activity. Now let's talk about the genetic / biological basis of McArdle. To start off, the inheritance is autosomal recessive. What this means is you need to inherit a mutated gene from both of your parents to be symptomatic. And to elaborate that a little more, if you get a copy that's mutated from one parent and a copy that's not mutated from another parent, you'll be a carrier with the possibility to pass down McArdle, but you yourself will not have it. And then the most common type of mutation that results to McArdle is on the R50X region of the PYGM gene, and the mutation that most commonly occurs is a nonsense mutation, which is the coding of a premature stop codon. And what this does is produce a non-functional protein. And then something that I'm going to touch on in a bit later is where the most reported cases come from, being Caucasian populations. And then going on chromosome 11, the PYGM gene has 20 exons. What an exon is, it's a part of a gene that is coded. There's a lot of parts of a gene which get spliced out, and only what remains is what's coded, and those are called the exons. And what these mutations all lead to is a failure in protein folding, protein stability, protein transport, and protein translation. Essentially, all the factors that lead to properly building and moving proteins don't work. And then going back, the most common mutation that leads to McArdle, the nonsense mutation on R50X, you can see the distribution on a world map, with the highest reported cases being from the UK, a predominantly Caucasian population. There's also some unknown, unrelated, or irrelevant aspects of PYGM in McArdle, that being the promoter region, repressor elements, and HOX genes. And I'm still going to go over these just to sort of help explain why it would be important to know the relevancy of these aspects. So first, the promoter region, it's an area of DNA which is upstream or before the gene of interest that allows the binding of any relevant proteins that help initiate transcription. Some proteins could be like RNA polymerase or transcription factors. Repressor elements, on the other hand, are sort of the opposite. They're DNA sequences that bind to repressor proteins. And what this does is it blocks, reduces, or fully turns off gene expression. And then lastly, HOX genes. It's just a group of very important genes that play an important role in developing different parts of the embryo. And again, all of these are either irrelevant, unrelated, or unknown to the genetic or biological basis of McArdle disease, but it's still important to know why. For the diagnosis of McArdle disease, the most optimal way to do that is a combination of physical, biochemical, clinical, historical, and genetic analysis. The forearm exercise test is a pretty solid way to measure lactic acid and ammonia levels, and the relevance of finding those specific levels of those chemicals is when you exercise, lactic acid will build up. However, lower levels will be shown in patients with McArdle, and when you have a lower level of lactic acid, you have higher levels of ammonia as a side effect. So seeing that combination of symptoms post-exercise is a pretty telltale way of knowing someone might have McArdle. And then for genetic testing, you need to get clinical grade tests. that check for mutations in the PYGM gene. DTC tests, or direct-to-consumer genetic tests, do not diagnose or test for McArdle. Those types of tests are the ones you can buy online or stores, like 23andMe, for example. But it's important to know that if you're trying to diagnose McArdle, those tests are not clinical grade, and they will not give you that diagnosis. In terms of managing symptoms for McArdle, it's important to know that that is the most you can really do with it, managing and reducing symptoms. McArdle is an inherited disease. It's genetic, meaning there's no cures or medicine for it. So the best you can do is try to reduce symptoms. But the first way to manage symptoms is regular aerobic exercise, which can improve your oxidative capacity. What that is, it's how well your muscles can use oxygen to make energy. And then focusing on that, improving your oxidative capacity with regular aerobic exercise can reduce the frequency of painful episodes. Other ways are having a high protein diet and genetic counseling. And then lastly, pre-exercise carbo loading, which can help reduce symptoms. However, there's a trade-off with that. It reduces your second wind, so that is a strategy that kind of varies depending on your situation. There's a lot of relevancy with genomics and bioinformatics in terms of McArdle. First, with the genomics, sequencing the PYGM gene and examining the whole exome of interest, we're able to identify and sequence the exact area of the PYGM gene that has the error. And then, as mentioned before, the R50X nonsense mutation that leads to the non-functional enzyme of McArdle is a big feat of genomics. Other relevancy to genomics is finding frequency among populations and then treatment and management of symptoms. Without genomics, we wouldn't have data banks that show global distribution patterns. And with genomics, we're able to conduct specialized testing to see who qualifies for precision therapy trials. Then there's also a lot of bioinformatics sites that can give us a lot of information and a lot of relevancy to McArdle. Interpro and Uniprot can help model how specific mutations in PYGM can disrupt enzyme activity. Sites like ClinVar, HGMD, and DBSNP can help differentiate disease-causing mutations from harmless ones. And then sites like the Human Protein Atlas and GTEx can show where PYGM is most expressed in like our bodies, being human skeletal muscle. This can lead to classifications of symptoms. And then lastly, PDB and Proteopedia can help generate 3D structures of proteins, and in this case, glycogen phosphorylase. And overall, these sites together can help pool together research and accelerate diagnosis and future research that we're getting by getting a good baseline on the essential information that you need. There's some relevant research that is done and that is to be done in terms of McArdle. Animal models such as mice, cattle, and sheep are being genetically modified to study McArdle disease. Some of these tests include genetic therapies, enzyme replacement strategies, and pharmacological efforts to go over nonsense mutations. And then just to kind of help explain pharmacological efforts, they're drugs that help ignore stop signals and mutated genes and make full proteins, which is extremely relevant to McArdle as the protein is produced, but it's in a non-functional one. And then increased screening is another way that is important in terms of research to McArdle. High rates in Caucasian populations might be only because of healthcare disparities in genetic screening. Low-income areas may have the same rates, if not more, just these cases aren't being reported. Thus, we don't think that they have high rates. So to conclude, McArdle is a rare autosomal recessive disease that causes immense pain in skeletal muscles. A single gene can have a huge effect on one's ability to be physically well. And with physical ailments, there's a chance of a psychological toll. While no cure currently exists, there are efforts to diagnose, reduce, and hopefully someday end symptoms. Awareness and education on the disease are key to lowering rates of delayed diagnoses, allowing therapies to start earlier. And as a send-off, this is the works cited.